Cloning vectors. So what is a vector? Vector is a DNA molecule, right? It is a DNA molecule that can carry a foreign DNA segment and replicate inside the host cells. So this vector molecule, it carry a foreign DNA segment. So it is called the vehicle since it carries, right? It is a vehicle DNA, or we can call it as a carrier DNA or carrier DNA. Okay. So the vector DNA is also called as a vehicle DNA or a carrier DNA because it carries the foreign DNA segment. Examples of the vectors are plasmids and bacteriophages. So we already discussed about the plasmids, that is, they are the autonomously replicating circular extra chromosomal DNA of bacteria. That is, they are not present in the chromosome. So they are the extra chromosomal DNA. We already know the structures, right? So this is the bacterial DNA and these are the plasmids. So this plasmids act as a cloning vector. So they are autonomously replicating circular extra chromosomal DNA of bacteria. But if you take some plasmids, they have only 1 to 2 copies per cells. They are present only in less number. So we can say it as low copy number. So copy number is the number of copies that are present, right? So others have 15 to 100 copies per cell. So it depends on the plasmids. And we know the structure of bacteriophages, right? So what are bacteriophages? They are the viruses which infect the bacteria, right? So this is the structure of a bacteriophage. So these bacteriophages, they have high copy numbers. That is, they are present in more number, right? So they have the very high copy number of their genome within the bacterial cells. So they are mainly used for the expression or the production of large number of the target protein, right? So when cloning vectors are multiplied, when these cloning vectors are multiplied in the host, for example, here it is a bacteria, right? So the linked piece of DNA is also multiplied to the numbers which are equivalent to the copy number of the vector. So if the vector is present, or the vector in the sense here, if it is a plasmid or the bacteriophage, if it is produced in 2 copies, right? So now the target DNA or the foreign DNA will also be produced only in 2 copies. But if it has a high copy number, so here the target DNA or the foreign DNA will also be produced in large number. So that is the advantage of having the high copy number. Okay. So here the bacteriophages have very high copy number of the genome within the bacterial cells. So we saw about the features that are required for cloning into a vector. We saw about the 4 features, right? So the first one is the origin of replication. So what is the role of origin of replication? So this is the cloning vector which is called PBR322. So this is an E. coli cloning vector PBR322, and it is an artificially synthesized cloning vector, and it was actually synthesized in 1977 by Bolivar and Rodriguez. So first PBR322 was the first artificial cloning vector which was constructed in 1977 by Bolivar and Rodriguez. Okay, so this is the first artificially constructed cloning vector. So here if you see the structure of PBR322, so what are the main characteristics of this PBR322? So what do you mean by this AMP R and TET R? That is, that represents the ampicillin resistant and the tetracycline resistant genes that are present, right? So they are the selectable markers, that is ampicillin resistance and tetracycline resistance. So the main characteristics of PBR 322 are: one is the selectable marker, and here what is all these represent? So these are all the restriction site, that is it has the restriction site for the restriction enzyme SAL1, BAMH1, HIN3, CLA1, EcoR1, and PBU1, and PBU2, and PST1. So this diagram is more important. Sometimes they may ask by giving A, B, C, and they may ask you to write what is A. For example, here instead of EcoR1, here they may give A, and they may give that CLA1 and HIN3. So what is the other restriction enzyme site which is present here? So we have to be clear with this diagram. So the PBR 322, it consists of the selectable marker, that is it contains the resistance genes for ampicillin and the tetracycline, and they have the restriction site for all these restriction enzymes, and they have the origin of replication, that is ORI. And what is this ROP represents? That is it codes for the, it is ROP, it codes for the proteins which are involved in the process of replication of plasmid, which are involved. Yeah, sure. Which one you didn't understand? Yeah, I'll explain. We'll see this diagram again. The proteins which are involved in the process of replication of plasmid. Okay, so these are the ROP. So this ROP or at that point, okay. So if you take the ORI, that is the origin of replication, yesterday itself we saw that that origin of replication is required for the replication to start, right? So the foreign DNA, they won't have the origin of replication, so they cannot multiply on its own. So the foreign DNA, it gets integrated with the vector DNA and then it is introduced into the host. So here this origin of replication is a sequence. So we are saying about this point. So this is the ORI, or the origin of replication. So these sequences is called the ORI, and here the replication starts. So a piece of DNA linked to ORI can replicate within the host cells. So whatever the sequence which is linked to the ORI, they can replicate. Okay, and another feature of this ORI is it controls the copy number of the linked DNA. So this ORI region, or the origin of replication, is responsible for the production of the copy number. What do you mean by copy number? It's what we saw. Some plasmids, they can produce only in 1 or 2 copies. But some plasmids, they can be produced in about 15 to 100 copies also. So how it is happening? Only based on the, that is this ORI. It is responsible for the copy number of the linked DNA. So whatever the DNA which is linked to this ORI region will be the copied. For example, if a particular, this plasmid is having the copy number of 2, the target DNA will also be copied only 2 times. But if the plasmid is having the copy number of about 100, that is if it is copied 100 times, then our target DNA will also be copied 100 times, right? So, but this is controlled by this ORI region. So this also controls the copy number of the linked DNA, okay? So for getting many copies of target DNA, so if you want to get more copies of the target DNA, it should be cloned in a vector whose origin support high copy number. So they are dependent on the sequences which are present in the ORI. Okay, is it clear? So this is the role of origin of replication, or the sequences in ORI region. Okay, so this region is responsible for the starting of replication, and this also controls the copy number of the linked DNA. So this is the artificially synthesized cloning vector, PBR 322. P, it represents the plasmid. BR, actually it represents the scientists, that is Bolivar and Rodriguez. They first constructed this PBR 322. Okay, so clear. So this structure is very important. You go through this and you must be clear about the even the enzymes sites and the ampicillin resistant, that is selectable markers, and ORI and ROP. ROP, it codes for the proteins which are involved in the process of replication of the plasmid. And also we have to be clear with the location. For example, this tetracycline resistant genes, they contain the recognition site for the enzymes BAMH1 and SAL1. Okay, so here, and the three restrictions sites, EcoR1, CLA1 and HIN3, and in the ampicillin resistant region they have the restriction site for the enzyme PVU1 and PSG1, and the sites are also very important. Okay, so next feature is selectable markers. So what is the role of selectable marker? So it is a gene that helps to select the transformants and eliminate the non-transformants. So what do you mean by a transformant and a non-transformant? That is, if a piece of DNA is introduced into a host bacterium. So now the DNA, the plasmid is introduced into the host bacterium, right? So first what we are doing, we are cutting the foreign DNA and it is aligned with the vector DNA, and this vector DNA is now introduced into a host bacterium. So that is called the transformation, right? Such a bacterium is called the transformant. Why it is called a transformant? Because it contains that plasmid with the foreign DNA, right? So it is called a transformant. And if transformation does not take place, it is non-transformant. If that plasmid is not integrated with the host, then it is a non-transformant. Okay. Selectable markers of E. coli. It includes genes coding for resistance to the antibiotics ampicillin, chloramphenicol, tetracycline, and kanamycin. So what are all these selectable markers? So in this example, you have the two resistant genes. One is ampicillin, another one is tetracycline. Similarly, we can insert the genes for resistance to chloramphenicol and other antibiotics also, right? So these are the selectable markers of E. coli. But if you take the normal E. coli cells, they do not carry resistance against any of this antibiotics. So this, we are introducing the resistant genes, that is ampicillin resistant and the tetracycline resistant genes here in PBR 322, right? For selecting the recombinants. So for selecting the recombinants from, or distinguishing the recombinants from the non-recombinants, we are introducing the genes which are resistant for antibiotics. Okay, so to link the alien DNA, so we need the cloning sites, right? So to link the alien DNA or a foreign DNA, we need the cloning sites. So what are these cloning sites? They are nothing but the restriction sites, that is the sequences. For example, we saw in the diagram, right? So here, if it is an eco R1, it represents it. Here the sequence will be GAATTC, which is the recognition sequence for eco R1. So the eco R1 can cleave at that site. Similarly, here CLA1 means here they have the recognition sequence for that particular restriction enzyme. And then here they have the sequence for HIN3. So similar, so these are the cloning sites. So what is the role of this cloning sites? So we can use this eco R1 enzyme for cleaving this DNA, and also the foreign DNA should also be cleaved with the same enzyme, right? So that it can be inserted. So yesterday itself saw the basic steps that are involved, right? So we need the recognition sequences for the particular restriction enzymes, or we need the cloning sites. Okay, so to link the alien DNA, the vector, it needs a single or very few recognition sites for restriction enzymes. So what do you mean by this? It needs only a single or a few recognition sites for restriction enzymes. For example, if you are treating it with the ECO R1, right? You are treating with ECO R1, if it has only one recognition sequence, it is in the whole of this plasmid vector. If it has only one ECO R1 recognition site, then only the cleavage will occur here. But if it has many ECO R1 site, what will happen? It will cause cleavage in multiple locations, right? So it causes the formation of more fragments of this vector, right? So we need to have only a single or a very few recognition sites for restriction enzymes. And more than one recognition site, it generates several fragments. So there will be formation of many fragments and it complicates the gene cloning. Then cloning will not be possible if there are many recognition sites, right? So ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistant genes. So where the foreign DNA is fused or ligated, it is at the restriction site present in one of the two antibiotic resistant genes. So why the ligation is carried out at that point? Because we can easily select the recombinants, whether the DNA is linked or not. We can easily find out if it is joined particularly at the restriction site and is present in two antibiotic resistant genes. Okay. For example, the ligation of foreign DNA at BAM-H1 site of tetracycline resistance genes. So where the BAM-H1 is present. So this is the BAM-H1. Here the BAM-H1 recognition sequence will be present. So if it is treated with BAM-H1, it causes a cleavage at this point, right? So it is present in the tetracycline resistance. So the BAM-H1 site of tetracycline resistant genes in the vector PBR322. So if ligation does not occur, if there is no fusion or the joining with the foreign DNA, it did not occur, it is called the non-recombinant plasmids. But if it is fused with the foreign DNA, then it is called the recombinant plasmid. Okay. So when a foreign DNA is inserted within a gene of bacteria, that gene is inactivated. So it is called insertional inactivation. Okay, so what do you mean by insertional inactivation? That is, if we are introducing the foreign DNA by using that restriction enzyme, that is BAM-H1, if the recombinant is present or if it is fused together, what will happen? Here the recombinant plasmids, they lose the tetracycline resistance due to the insertion of foreign DNA. So we are able to follow this. For example, we are treating it with BAM-H1. So this is the vector. Now the foreign DNA we are going to introduce into this vector DNA. Then the site, that is BAM-H1. So we are treating it with the enzyme, restriction enzyme BAM-H1, and also the foreign DNA is treated with the BAM-H1, right? So both are cleaved with the same restriction enzyme. So here, if there occurs an insertion of the foreign DNA at this site, it is at the site of BAM-H1, what will happen? This will become inactivated. That is, tetracycline resistance will be inactivated due to the insertion of foreign DNA, right? So here, the recombinant plasmid is nothing but the foreign DNA is fused now with the vector plasmid, right? So then it is called a recombinant. So the recombinant plasmids, they lose tetracycline resistance. Actually, they are having a tetracycline resistance, but here they lose the tetracycline resistance due to insertion of foreign DNA. Clear? So when these plasmids are introduced into E. coli cells, so 3 types of cells are obtained. We will get 3 types of cells. So